Hello, everyone. I'm Miles Skoda, and this is the OutsourcePharma.com virtual event series. And today we're talking liquid phase peptide synthesis, regulatory, analytical, and QC perspectives. Now, joining me for this presentation are two industry experts from Snapdragon Chemistry, a Cambrex company. First, Dr. David Wang, Senior Director of Analytical Science, and Dr. Adrian Amador, Director of Chemistry, who together are going to cover an introduction to LPPS and its applications. They'll look at the regulatory landscape for peptides, discuss impurity ID, and go over chiral starting material control and chiral diastereomeric impurity control. So we will be holding a Q&A session as well following this presentation. So please feel free to submit questions as we go along. We'll get to as many as possible during that Q&A, and any that we don't get to, I'll pass along to the team here for potential follow-up after the event. And then just a bit of housekeeping. If you run into any issues today, please try refreshing your browser first. If your problem persists, you can submit a question through that Q&A, and I'll help get you back on track. And then finally, this event will be available on demand, usually within a day, often much less. You'll get an email with a link to it, or you can just use the same link that you used today. So with that said, let me turn it over to Adrian to get started.
Thank you, Miles, for the great introduction. So happy to kick this webinar off to discuss some of the regulatory, analytical, and QC areas for peptides in general, and more specifically for liquid phase peptide synthesis. So first, a brief outline of the structure of this webinar. We'll start with an introduction to Cambrex as a whole and Snapdragon specifically, a brief introduction to liquid phase peptide synthesis and how we think about this here at Snapdragon. And then I'll pass the baton over to David Wang so that he can introduce peptide analytical chemistry, go into the regulatory framework around peptides, approaches for identifying and characterizing peptide impurities, chiral purity controls, and then finally to conclude the talk. So first, an introduction to Cambrex. So Cambrex is a global contract development manufacturing organization situated in the United States and Europe. We cover a range of services from analytical services to drug substance and drug product with several relatively large manufacturing sites in both the US and Europe. Snapdragon Chemistry specifically is situated in the greater Boston area in Waltham, Massachusetts. And we focus largely on process development and early phase deliveries under manufacturing. And we also have a special focus in the complex synthetics, including peptides, oligonucleotides, and synthetic polymers, as well as continuous flow technologies. So across Cambrex, we offer a number of services from early development services. It includes analytical, standalone analytical testing, solid state characterization, and stability storage and testing, as well as some drug product services. In the drug substance services area, we offer GMP, API, and non-GMP manufacturing, a whole suite of process development, characterization, and validation services, analytical R&D and validation. And we try to focus where possible on sustainability in our manufacturing approaches. And as mentioned on the previous slide, we have significant expertise in a number of specialized technologies, including the handling of high potency compounds and controlled substances, continuous flow, R&D and manufacturing, the complex synthetics landscape, including peptides, and biocatalysis development and manufacturing. Okay, so to jump into introducing liquid phase peptide synthesis and how we think about this here at Snapdragon and Cambrex as a whole. First, just to kind of briefly summarize why we think some of these more sustainable manufacturing approaches are necessary. And as many people attending this webinar will be very well aware, peptides have become over the last couple of decades a major modality for drug discovery, and demands continue to rise and now are on the order of metric tons. Traditionally, standard solid phase peptide synthesis is the go-to technology for both the discovery stage as well as manufacturing. And however, this technology was originally designed for the discovery in low kg scale quantities. And so with that, it tends to have a relatively large environmental impact. However, particularly with the introduction of the impact of peptides in the metabolic space, the increase in demand for peptide therapeutics continues to rise. And so the need for capacity also continues to increase. So liquid phase peptide synthesis, when and why would you utilize this type of approach for manufacturing peptides? So from our perspective, for early phase applications in low peak annual demands, SPPS is still generally the technology of choice, just considering the simple approach to go from development up to manufacturing. It tends to be relatively plug and play. There's a lot of automated systems to do this, and it also potentially has reduced labor costs and plant times just due to the kind of simple nature of this technology. So liquid phase peptide synthesis, conversely, is really useful for much larger scales and when much higher peak annual demand is needed, and it offers several advantages that make this the case. One of these major advantages from our view is the use of standard batch reactor technologies, where there's greater available global capacity just due to the types of reactors that are able to be used for this. Another advantage is from the chemistry point of view, where you have improved reaction kinetics. It's all solution phase chemistry. You don't have to have your reagents penetrate within a resin matrix. So this can provide improved coupling efficiency, which can often lead to reduced stoichiometries being required. And there's also drastically improved volumetric efficiency overall, and this can result in drastic reductions in the overall process mass intensity when you're using this type of technology. And I have just an image on the bottom right of this slide showing the relative PMI for liquid phase processes compared to solid phase processes, where you can often get orders of magnitude reductions in your PMI through the adoption of an LPPS process. So from our perspective, when the material demands are relatively high, or there's a strong mandate for reducing your process mass intensity, LPPS can be well worth the increased development time that's required in order to implement a process like this. So at Snapdragon, we tend to focus on a total solution phase peptide synthesis rather than a hybrid route. And this utilizes a fragment coupling approach, where in liquid phase, you would be generating generally shorter fragments of roughly 10 to 15 amino acids. And then again, in liquid phase, these fragments would be brought together with liquid phase fragment condensation to bring you to your full length peptide, which can, it's been demonstrated for greater than 40 amino acids relatively regularly. So this can offer improvements in quality, allow for parallel production, reduction in the overall risk due to breaking this up into smaller fragments, and also improved supply chain management by being able to do this in standard reactors, break up the fragments maybe between different manufacturing sites, and then bring them together at a later date. So kind of looking at the overall process, there's a lot of parallels between LPPS and SPPS. The general amino acid activation, coupling, and deprotection cycle is identical to how this is performed for SPPS. The main difference is that this is all in the liquid phase. And rather than washing away the byproducts and excess reagents from a resin, this is performed with liquid-liquid extractions as shown on this slide, where you would have, during your deprotection, you'd be adding a base to, in the case of FMOC chemistry, or maybe a hydrogenation in the case of CBZ chemistry. And then if you're doing FMOC chemistry, you would have a scavenger in it as well. Once deprotection is complete, then you would wash to remove excess base and byproducts from the deprotection, proceed to activate your incoming amino acid and couple that, and again wash to remove excess reagents and amino acids. So again, the advantages for LPPS is that it provides the use of standard reactors, higher reaction concentrations, which improves productivity and volumetric efficiency, reduce the need for excess reagents, reduce solvent demand, and more simplified analysis, as you can simply sample your organic phase and analyze this by HPLC or UPLC. So going through the overall stages of an LPPS process, this is laid out on this slide here. Again, a lot of this is analogous to SPPS. So you have your initial raw materials coming into the process. For tag-assisted peptide synthesis within LPPS, you would have a C-terminal esterification where you install a hydrophobic tag to either a single amino acid or maybe a di or tripeptide. You would then enter the standard peptide elongation sequence, which is very similar to SPPS process, where you have deblocking to remove your FMOC or CBZ group, amide coupling, and then you repeat this until you get to the length of your fragment or peptide. Once peptide elongation is complete, you would then proceed to your hydrophobic tag cleavage to remove the tag off your C-terminus. And then if this is a fragment condensation to build a larger peptide, you would then have solution phase fragment coupling, and then finally isolation of your full length peptide. So going step by step, looking at the quality risks that are experienced throughout the LPPS process. Within raw material sourcing, you have to worry about the purity, potency, and chiral purity of the reagents that you're bringing in, particularly the amino acids. Within tag installation, there's a few risks introduced here. This tends to have a higher risk of racemization than a standard amide coupling through this esterification. So any chiral amino acid, you have to worry about epimerization of your C-terminus. If you're using basic conditions to install that ester, there is the risk of endocoupling. If the reaction conditions are a little too intensified, you can get in situ deblock of your FMOC group, which can result in double coupling, which would be purities that are carried throughout the synthesis. And then you also have to worry about residual tag being present, as this can introduce downstream deletion impurities through esterification that can happen throughout the introduction of new amino acids. During the deblocking step, you have to worry about conversion, which can introduce deletion impurities. If you're doing FMOC chemistry, you'll generally be using a base to remove that FMOC group. And so multiple byproducts can result from the exposure of a base to a growing peptide chain. Two of those are shown on this slide, one being diketopiperazine, as shown in the scheme to the right, as well as impurities through aspartamide or glutaramide formation. If using CBZ chemistry, deblocking will generally utilize hydrogenation, and then you have to worry about sometimes you can observe side chain reduction. For example, reduction of tryptophan has been observed in the past under hydrogenation conditions. During the amide coupling step, again, racemization is a major concern. So keeping track of these and having an appropriate approach for analyzing the introduction of diastereomeric impurities is pretty critical. Incomplete coupling needs to be avoided in order to avoid deletion impurities, as well as avoiding double addition impurities, particularly if you're using basic coupling conditions where you're using a base such as diisopropyl ethylamine or similar, as well as incomplete purge of residual amino acids, which can result in double addition impurities. The tag cleavage step, the types of risks here depend on the type of hydrophobic tag that's being used. So if you're using an aliphatic ester, generally you'll need to use saponification to cleave that tag. This can result in epimerization of the chiral centers along your peptide chain, particularly of the C-terminus, as well as side chain deprotections, for example, of terp butyl esters can occur under basic conditions. If you're utilizing a benzylic tag, this can be removed under hydrogenation. You have to worry about residual metals being present in your final peptide, as well as mentioned on an earlier slide, side chain reduction. 2,4-dialkoxy benzyl tags, these are generally removed with dilute acids. And the main impurity risk here is side chain deprotection, as most of the side chain deprotections are generally removed under acidic conditions. And then finally, allylic tags, which are often removed under palladium allyl chemistry conditions with some type of reductive scavenger. Again, residual metals have to be considered, as well as the propensity for allyl group transfer to other nucleophilic functional groups present in your peptide. And then moving forward to the fragment coupling. During downstream fragment condensation, C-terminal epimerization is a critical risk here. So the use of appropriate coupling agents, the appropriate temperature, and identifying an appropriate disconnection point to for your retrosynthesis for where you're going to combine your fragments is critical to reduce that C-terminal epimerization here. And then finally, isolation, a very critical stage in the overall process, where you have to worry about all of the general residual impurities that would be tested in any standard QC release, such as residual solvents, residual metals, water content, potency, aggregation, dimerization. So overall, due to the complexity of the peptide synthesis process, which is present for both SPPS and LPPS, as well as the unique challenges of LPPS, very robust analytical techniques are needed to ensure the overall quality of your final API. So with that, I will pass the baton over to David. Thank you.
Thanks, Adrian. Now let's move on to the analytics. We will begin with the analytical difference between SPPS and LPPS from analytical perspective. So the analytical strategy differs between SPPS and LPPS because of their distinct impurity profiles. And SPPS mainly generates truncates and deletions, give a relatively cleaner crude product with lower racemization risk. So HPLC, LC-MS is generally sufficient for SPPS analysis. In contrast, LPPS produce more complex crude mixtures and a broad impurity profile, as Adrian mentioned earlier, including racemization, tag byproducts, and dimers. This demands a more comprehensive toolbox: LC high-resolution mass spectrum, derivatized LC-MS, SFC, CE, and NMR, and stricter impurity identification and qualification, typically down to 0.5 to 1% of the impurity. So more broadly, peptides are a growing therapeutic class, and their complexity require us to manage a unique challenge, like sequence-dependent impurities and degradation pathways. So the goal is really ensure identity, purity, potency, and safety, generate robust data for eventually regulatory filings. So when we analyze peptides, three big challenges always comes up. First is structure complexity. Peptides aren't just simple chains. They can have sequence variations, chemical modifications, and charge and size variance. Each of these adds layers of complexity that small molecule methods cannot fully capture. Second is stability. Peptides are prone to deamidation, oxidation, aggregation. These degradation pathways can impact the potency and the safety, so they must be carefully monitored. Finally, there are analytical limitations. It's not always easy to separate isomers or detect early degradation products with enough sensitivity. This means we need to be very deliberate in choosing orthogonal methods to get the full picture. Together, those challenges explain why peptide analytics require specialized strategy, not just standard approaches borrowed from small molecules. Since not everyone may be an expert in peptide chemistry, let me start with some quick background before we dive into the analytical details. So at the core of peptides are amino acids, each built around a central carbon with a variable R group that defines its property and drive structure diversity. Another key feature is chirality. Most amino acids exist in L and D forms, but the biology almost always uses L form. This chirality is important because it affects folding function and how regulators expect us to measure and control stereochemistry. Understanding those basic building blocks is the foundation for peptide analytics. So here we can see the 20 standard amino acids, the essential building blocks of peptides and proteins. What makes them different is their side chains, or R group. We can group them into categories. Non-polar R groups, those are hydrophobic and driving folding into the protein core. Polar and charged groups, they contribute to solubility, hydrogen bonding, and ionic interactions. Aromatic groups like phenylaniline, tyrosine and tryptophan are bulk and can absorb UV light, which we use analytically. Those chemical differences aren't just academic, they directly affect the peptide stability, solubility, and how we analyze them. In short, that group defines both structure and function. So now let's move to the regulatory framework. One of the first regulatory questions with peptide is, where do they belong? Small peptides, typically under 40 amino acids, are often treated like small molecules on ICH Q6A specification. But once they get larger or more complex, especially recombinant peptides, the expectation shifts close to biologicals on ICH Q6B specification for biologicals. In practice, regulators expect us to characterize impurity down to 0.1 to 1%, a range reflected in European pharmacopoeia monographs and reinforced in the EMA 2023 draft guidance for synthetic peptides. Beyond that, we must monitor stability risks like deamidation, oxidation, and aggregation, and demonstrate control over identity, purity, and sequence variants. So when it comes to stability testing, peptides behave very differently from a small molecule. For small molecules, we mainly think about hydrolysis, oxidation, and traditional HPLC UV is usually enough to track degradation. But the peptides are much more sensitive. They undergo sequence-specific degradation like deamidation of asparagine and glutamine, oxidation of residues such as methionine, tryptophan, and cysteine, and they can aggregate. That's why peptide stability study can't rely on small molecule method alone. We need a tailored LC-MS approach, and the shelf life assessment must confirm not just potency, but also sequence integrity. The key takeaway, peptide stability testing has to go beyond HPLC UV. LC mass spectrum with sequence integrity is essential. So, as you mentioned earlier, the challenge for liquid LPPS mainly about impurities and the chiral control. So now let's move to the peptide impurities. So there are a few categories of impurities. First is synthetic related impurities. Those are things like truncated or deleted peptides, incomplete deprotection, sequence isomers, or racemization leading to D amino acids. Second, process-related impurities. Residual solvents and reagents can carry through from manufacturing. And third, degradation-related impurities. Oxidation of methionine, tryptophan, or cysteine, deamidation of asparagine to aspartate or isoaspartate, hydrolysis, backbone cleavage, or aggregation into dimers. So overall, impurity is not just one-dimensional. They come from synthesis, process, and degradation. Each needs to be identified, controlled, and monitored to meet regulatory expectation and ensure product safety and quality. So when it comes to the impurity identification, we rely on toolbox of complementary technologies. The primary workforce is LC, high-resolution mass spectrum, which gives us accurate mass and sequence mapping through mass analysis. For chiral analysis, HPLC or GC, after hydrolysis and derivatization, allows us to distinguish D from L amino acids, critical for regulatory compliance. We also bring in orthogonal methods like CESDS, capillary zone electrophoresis, NMR, FTR, or CD to get insight into charge variance and conformation. And finally, peptide mapping using enzymatic digestion plus LC-MS lets us pinpoint site-specific modifications. The key point is no single method is enough. impurity ID requires an orthogonal toolbox. So here is the case study of impurity we see during LPPS. On the left, you can see the HPLC method using C3 column with TFA, water, and Sinatral, 15 minutes run at column temperature 40 degrees. This setup gives us good separation of peptide fragments. On the right is the chromatograms. The peak you see here, P1, P2, P3, represents different peptide fragments, typically 5 to 15 amino acids in length. And these fragments are intermediates from liquid LPPS synthetic work. So this slide shows an overlay of chromatograms highlighting different impurity signal in peptide synthesis. You can see distinct peaks labeled for the peptide itself, but also for impurities such as residue protecting group like FMOT, tags, stabilizers like BHT, and other process-related byproducts. The power of this type of analysis is comparison. You can clearly separate and identify where each impurity originates. That helps us not only clean up the process, but also build a strong impurity profile for regulatory filings. The key takeaway is that overall, overlay chromatograms give us a clean picture of both desired product and unwanted byproducts, which is essential for process optimization. So here we're looking at a semi-preparative LC-MS used for peptide purification. On the chromatograms, you can see 6 fractions being collected. The main peptide peak is isolated, while surrounding fractions captures impurities. The workflow is straightforward. First, collect fractions. Second, characterize them with LC-MS, GC-MS, and NMR. Third, confirm purity and assign impurities. And finally, track fractions until regulatory quality IDs are confirmed. The key point is that this approach not only isolates the desired peptide, but also systematically characterizes impurities, building the traceability needed for regulatory filings. So this slide shows how we use preparative LC-MS to characterize purified fractions. At the top, you see the crude peptide, which is a mixture of product and the multiple impurities. By fractionating and analyzing each peak, we can separate out the true peptide from low molecular weight impurities, epimers, and high molecular weight byproducts. For example, fraction 2 gives us the main peptide, while fraction 3 to 5 reveals epimers and the major impurity. Each of these fractions is then characterized further using LC-MS, GC-MS, and NMR. So we can confirm purity and assign each impurity with confidence. So the takeaway is that the preparative LC-MS is not just purification. It's a powerful way to build a complete impurity profile that meets regulatory expectations. So now let's move to another key area of the peptide analytics, the Chiral purity controls. So this slide shows the overall workflow for Chiral and the impurity analysis peptides. On one slide, we analyze the intact peptide. On one side, we analyze the intact peptide with LC high-resolution mass spectrum, or LC-MS to profile, map, and check stability. From there, we can apply Chiral SFC-MS as a fast orthogonal and even preparative tool. On the other side, we can hydrolyze peptides to amino acids, then use derivatization methods like MOFI or FTAA to separate enantiomers. This allows Chiral LC-MS considered the regulatory gold standard for DL ratios, and the Chiral GC-MS, which provides very high enantiomer resolution and serves as confirmatory approach. The key message here is that there is no single method for Chiral and the impurity analysis. Instead, we use a workflow or complementary tools with LC-MS, MS as the foundation and orthogonal Chiral methods to ensure regulatory confidence. And the supercritical flow chromatography, or SFC, uses CO2 in a supercritical state at the main mobile phase. It combines the speed of gas chromatography with the versatility of liquid chromatography, and it's full compatible with detects like the UV, mass, and the ELSD. So when do we use it? SFC is especially valuable for Chiral separations, for providing orthogonal conformation in regulatory filings, and for purifying impurity that the LC or GC cannot resolve as cleanly. So an added advantage is the sustainability. SFC is greener and faster than traditional LC-GC because it uses much less organic solvent. The key takeaway, SFC is not a replacement, but a complementary tool, giving us speed, resolution, and regulatory confidence. So everything has two sides. It also has limitations. And so it requires specialized instrumentation and expertise. It's less standardized than LC in terms of regulatory guidance, and highly polar analytes often need modifiers to be resolved effectively. So this case study focuses on Chiral purity in peptide synthesis. When we check Chiral purity, the process starts with raw materials. Using SFC, we measure the inherent D isomers content already present in amino acids themselves. Next, we account for racemization introduced during synthesis or processing. Finally, we must be careful to avoid artificial D isomers created during analysis. That's why we use deuterium chloride hydrolysis instead of hydrochloric acid. DCl stabilize steric centers during acid hydrolysis, where HCl can promote racemization and create false D isomers. By using DCl, we preserve the trace stereochemistry of amino acids, so the D isomer measured reflects only what comes from raw material and process-related racemization, not from the test itself. So the key point here is the D isomer can arise from three sources, and raw materials, the process, and analytical artifacts. And our methods must distinguish between them to give regulatory confidence in the data. When it comes to Chiral purity, there are two main LC-MS approaches, direct or derivatized. Direct LC-MS is fast, require minimal preparation, and is great for quick screening or as an orthogonal check. With the right Chiral columns, you can resolve D and L isomers directly. Derivatized LC-MS, on the other hand, is more involved. It requires sample preparation and carries from risk of artifacts, but it's also the regulatory gold standard. It provides the most reliable D and L ratios, works better for polar amino acids, and is required for release testing. So here is the side-by-side comparison of direct versus derivatized LC-MS results. On the left, the direct LC-MS shows clear resolution of D and L isomer for amino acids like phenylalanine, serine, and vinyl. It's fast and effective for screening. On the right, the derivatized LC-MS use FDA derivatization, shows improved separation for more challenging amino acids like tyrosine, tryptophan, and histidine. This is why it's considered the regulatory gold standard. The validation metrics at the bottom confirm strong linearity and acceptable recovery in the 70 to 130% range. So the key message here is that direct LC-MS gives speed and orthogonality, while derivatized LC-MS provides the regulators expect for release testing. So in practice, we use a two-tiered approach. For development and support, we rely on direct LC-MS. It's fast, requires minimal preparation, and is ideal for screening and orthogonal checks. But for QC and release, we move to validated derivatized LC-MS method. This is what regulators expect. It provides robust DL ratios and amino acid count with validated accuracy. Together, this workflow balances speed in development with compliance in release, ensure both efficient progress and regulatory confidence. So here's the conclusion and the key takeaways. To wrap up, peptide demand tailored analyticals because of their complexity and a unique degradation pathway. Our primary tool is LC high resolution mass spectrum, supported by orthogonal method when needed. For chiral control, derivatized LC-MS remains the regulatory gold standard, even though direct methods are useful for support. Importantly, impurity above 0.5 to 1% must be identified and qualified and per the guidance. And finally, for QC and release, validated derivatized LC-MS combined with LC high resolution mass spectrum ensures regulatory compliance and confidence. So the big picture is faster tools drive development and the validated tools secure approvals. And to stay aligned with the global expectations from regulatory guidance, there are three key principles. So first, we use our orthogonal platform, multiple complementary methods to ensure full characterization. Second, we always align with ICH, FDA, and EMA guidance. So our data meets regulatory standards worldwide. And third, we apply validated phase-appropriate methods. That means using the right level of rigor in early development and a fully validated method for QC and release. So the bottom line is this approach ensures scientific robustness and regulatory confidence at every stage of development. So this brings us to the end of today's session, where we have worked through the analytical and regulatory challenges for peptides. Adrian worked through the synthetic part LPPS. And the entire strategy we talked here ensure both scientific rigor and compliance. Thank you all for your time and engagement today. We hope these insights were helpful. Now we're happy to take your questions. Thank you.
Amazing. Yeah, David, Adrian, thanks so much. This was awesome. We do have a lot of questions rolling in, a lot of questions. So if you do have some more questions, please get them in. We'll get to as many as we can here, and then we'll pass along the rest of them to the team for some potential follow-up after the event as well. And so, yeah, there should be the Q&A box on the side. You can submit those questions through. And then in addition, audience members, if you wanted to reach out to either of the speakers directly, if you check out the speaker bio panel over on the right side of your screen, click on the little blue circle below their picture, and that should let you just contact them directly if you had something maybe more specific or you just wanted to reach out to them and ask directly. But let's get into these questions here. And so I'm going to start with this one. So what advantages does liquid phase peptide synthesis provide over a traditional solid phase peptide synthesis?
Yeah, thanks, Miles. I can address this. Yeah, so kind of as laid out in the first portion of this webinar, it's largely from an efficiency point of view, as well as the manufacturing capacity that's available globally. So from our experience, SPPS can generally have a process mass intensity on the order of over 1,000 per amino acid that's being introduced. And a lot of the LPPS processes that we introduce is generally less than 100 per amino acid that's being introduced. So a drastic reduction in the amount of waste that's being produced. And then the amount of peptide per given volume within your reactor is also substantially greater. For example, we've produced batches of peptide on the order of 5 to 10 kgs within a single batch, utilizing just a 100 to 150 liter reactor, which we require a substantially larger reactor in order to do the same type of process with SPPS. And then when that process needs to be scaled further, this can go to any standard 16,000 liter vessel that's available globally at many different CDMOs or CMOs.
Awesome. Yeah, thanks, Adrian. Let's see, another one here. So when and how do you use GCMS for chiral impurity control?
Yeah, okay, I will tackle this one. So as we know that the GC, to be analyzed by GC, the material has to be volatile and also thermostable. And most amino acids actually do not meet those kind of requirements. So GCMS application here for LPPS usually is after hydrolysis and derivatization. So it offers a high enamel resolution for volatile derivatives. So as we know that some of the LPPS intermediate we have tag on it, which we also can derivatize specifically with some reagents that make the volatile. This is not primary for QC release tool, but the GCMS serves as a confirmatory or signal method for regulatory submission, comparability, or when LC-based methods are insufficient. So it really is a case by case, and mainly for supporting, not really a tool for release testing for LPPS process.
Perfect. Yeah. Thanks, David. Let's see. So again, thanks for those two questions. This next one's from Kian. So how should analytical specifications be set for peptide analysis?
Yeah, I also tagged this one. Yeah. So specification actually aligns with the ICHQ 6A and B, as I mentioned in the presentation that peptide is very unique. It's not complete small molecule, it's not biologicals. And so therefore we need to consider both of those specification guidance. So, but regardless, we should cover identity, purity, potency, and safety. And for impurities particularly, and we're talking about anything about 0.51% must be identified and qualified. And this actually comes from a EMA 2023 draft guidance. The guidance is still drafted, but this actually comes from earlier version with the EU EU compendia monographs. So basically, the industry transitional current requirement is 0.5 to 1%. And other than that, stability testing must include the peptide specific degradation study, and spec also involves the phase appropriately. So same as all those small molecules and biologics, full validation is expected by phase 3 and before NDA, BLA, and all those methods should be fully validated and to support specification setting.
Okay, yeah, good stuff. Let's see, another one here from Hannah. So how do you conduct ID testing to confirm peptide sequence?
Yeah, I also tagged this one. So ID testing, usually we use LC, high resolution mass spectrum. This is primary tool for ID testing. And this to confirm intact mass. And peptide mapping also, we can use the enzymatic digestion. There are many enzymes we can select to select the cleavage specific amino acids. And from there, then plus LC mass analysis, we can provide the sequence coverage and modification information. And our signal confirmation can be done by NMR, amino acid analysis, or even adenine degradation for short peptides. So yeah, there are many ways to do ID, but for regulatory requirements that at least 95% of sequence should be covered, should be confirmed, and for complex peptides.
Awesome. Yeah. Thanks, David. Let's see. Let's get another one here. Rapid fire these in. So what phase appropriate validation approaches do you follow for these peptide methods?
Yeah. So this phase appropriate development is similar to a small molecule category, and maybe also apply to biologicals. For preclinical phase 1, we really need fit for purpose qualification and for analytical methods. And we need to focus on the specificity and the sensitivity, and we can provide accurate information for assay for the main compound. And the phase 2, we start more focus on impurity. We need for the method, we need to have a precision. We need to consider reproducibility and their stability testing going on. So we need to consider the method should be stability indicating. And for phase 3, and that's really focused on the being preparation for the filing commercialization. So full ICH-Q2 validation needed to be compliant, and that require precision, accuracy, linearity, robustness, whatever outlined in ICH-Q2. So for peptide itself and the derivatized LC-MS, high resolution mass spectrum must be validated to regulatory standards. That's a little bit challenging part. And then we talk about the life cycle, post-approval transfer with compatibility studies and continued excellence development. So yeah, that's following the our other concepts, small molecule and biologicals.
Great, David, thanks. Why do peptide molecules need tailored analytical strategies?
Yeah, as we mentioned that the entire slide, and Adrian also mentioned for the synthetic work, and we use them in the LPPS. We use the different tags, and some tags are very hydrophobic, some are really hydrophilic, and they're all different purpose, and also there are different chiral impurities. And the structure itself is really complex structure. It depends on the length of the chains and how many amino acid units. So therefore, that impurity profile is different than small molecule. Right? You have a sequence-related impurity. You have here stereochemistry control, and standard small molecule assay usually just focus on one single molecule. But here we're talking about polypeptides. So yeah, therefore the strategy involved heavily on LC, high resolution mass spectrum, and the chiral control with different complementary methods. And so to ensure the identity, purity, potency, and safety. So basically the tolerant analytical strategy is specifically due to the nature of the molecule itself.
Great, thanks, David. Let's see, got time for a few more here for sure. So let's go. What are the advantages of supercritical fluid chromatography? And they're asking, when would you apply it?
Yeah, so FFC this offers very fast separation, and it can provide strong chiral resolution and low solvent consumption. It's green, right? So it's really reduced food carbon footage. And but it's I think a limitation we mentioned, we touched up in the presentation, that need actually a molecule to be hydrophobic, less polar to medium polar. So some amino acid actually cannot meet this requirement. Therefore, it also need a derivatization. So, and also it needs specialized equipment and less standardization across QC labs. So it's really good for early development to get quick results, and for apply for some peptide sequences, and mainly used for direct amino acid after derivatization to see quickly control the DL amino acids ratio. So yeah, it's not standard release testing, but good for orthogonal analysis and early development.
Great, thanks, David. All right, another one here. So this one's from Lucas. He's asking, so how do you control cysteine, cyclization, and oxidation with LPPS versus SPPS?
I can tackle this one. So in many ways, it's going to be very similar strategies between LPPS and SPPS, which is the use of appropriate protecting groups Typically, this is a trityl protecting group on a cysteine residue. It's going to be the same with SPPS and LPPS. One of the main differences, particularly if you're intentionally trying to add disulfide bridges within your peptide sequence, is going to be the staging of this. Oftentimes, the cyclization for SPPS can be done while still attached to the resin, whereas depending on the tag that you're using, you may want to cleave your tag with LPPS before you do your cysteine cyclization, as oftentimes the conditions that are used for that are not going to be compatible with the tag, particularly things like allelic tags and similar. So the staging of that and how you go about it can be quite different. And since this is a solution phase for LPPS, you also have to worry in maybe slightly different ways about dimerization and oligomerization if you're trying to introduce disulfide bridges. And so you might need to kind of stage the order in which you add your reagents with a reverse addition so that you're maintaining a very low concentration of the free thiols so that you don't end up dimerizing or oligomerizing your peptide.
Great. Yeah, thanks, Adrian. Okay, another one here. So as it is not feasible to have reference standards for each impurity, is area % okay for regulatory, provided we have a solid rationale to estimate similar detector responses for similar impurities?
Yeah, I will tackle this one. Yeah, this is very common, that very difficult to have a reference standard at the early stage for impurities particularly. And all the regulatory finding really is based on risk-based justification. So if you have a good justification, that's probably okay. But I think that justification has to be really strong. And I assuming that this area % is for LC analysis, right? And if you don't have RF, probably you need to have orthogonal method to adjust and what's the y percentage area counts give you the accuracy. And considering that UV chromophore is different from one to another one. So I guess that here, if you have a solid justification with orthogonal methods and you have your study, you have your report, and that should be okay. And eventually that probably you will have RF calculation. You will get, as the phase moves to the later stage, you will have all the information later on. So that's my perspective on this.
Great. Yeah, thanks, David. Let's see. So I've got one here from Cassie. So what is your approach for determination of epimerization impurities for those amino acids that cannot withstand the hydrolysis process involved with the deuterium derivatization process?
Yeah, this is the challenge topic. That's the reason that we mentioned the different methods. None of them can cover all the amino acids, and therefore some hydrolysis may not be a good tool for us to do it. Therefore, that we have to think about different way to do it. Maybe just use direct analysis with some software can help you to compare the peak without using those hydrolysis wet chemistry, and along with other technology that if we can analyze, as we mentioned, maybe we can analyze through direct derivatization without hydrolysis by SFC, and if the hydrophobicity is okay, and some of those maybe we can derivatize with make it volatile without hydrolysis with GC analysis, and so some has to be a combination of all those methods together for particular analysis of impurities. Yeah, there's no one answer to all of them. And that's the reason we talk about it. We need to have a common strategy. We need to have a combination of the different methodology to work on this. We did have such questions happening of us. So we just work together, use a combination of those tools and try to get the best solution we can.
Perfect. Yeah. Thanks again, David. Let's see. Maybe we can get to one or two more here, maybe two more here at least. So in your experience, have regulatory health authorities required chiral amino acid ratio determination on release for all batches or only for characterization?
So yeah, from my experience, those chiral analysis ratio is not expected for release testing. It's more a characterization study. So as long as you have this well controlled, and you demonstrate during the validation batches you are able to control well. So this will not be not a commercial QC testing. It's complicated more than the, usually it's difficult for QC to handle. And also, as long as you have a robust process, you can control the well. You demonstrate that during the development, this is not expected as a QC release testing. So short answer is it's a characterization study.
Great, thanks, David. All right. Let's get one more in here. And before I do, just a couple reminder for everyone, in the bottom right corner of your screen, there's just a great resource on what we talked about today. For download, just click there. It'll give you the document right away. And then again, you can reach out to our speakers directly on the right side of your screen, right next to their picture, little blue circle with the mail icon on it, and get in touch with them. And any more questions you can still ask through the Q&A, and I'll pass those along to the team. And yeah, let's get to the next one here and our last question. So what processing conditions present in SPPS require different reactors versus LPPS?
Yeah, so this is largely just the synthesis stage. SPPS, by definition for how it's typically done in a batch SPPS, requires you to have a large frit on the bottom of your reactor so that you can separate the liquid components from your insoluble resin. Whereas LPPS is just done in standard batch reactors the majority of the time, at least the way that we carry it out with just standard liquid-liquid extraction for the workup after each stage. Most of the upstream and downstream stages are going to be very similar between SPPS and LPPS, with things along the lines of the prep chromatography, lyophilization, if that's what's being used, or otherwise precipitation. It's going to be very similar between the two.
All right, Adrian, thanks so much. I think this is going to be a great place for us to wrap up. And so again, thanks again to Dr. David Wang, Dr. Adrian Amador. Appreciate you both being here today. This was a great presentation, and we got through a lot of questions. And audience members, thank you so much for all the questions, and again for joining us as well. Any other questions, reach out to our speakers on the right side of your screen. But with that, we do hope that you all have a great rest of your day, and we'll see you next time. Bye, everyone.
Thank you. Bye.